Hi, everyone. Happy Leap Day. I don't get to say that so often. So welcome to the Sentinel Innovation and Methods Seminar Series. My name is Judy Morrow. I'm an assistant professor in the Department of Population Medicine at Harvard Medical School and Harvard Pilgrim Healthcare Institute, and I'm also at the Sentinel Operations Center. A little bit about Sentinel to begin. The US FDA's Sentinel system helps to address questions related to the safety and effectiveness of medical products. Within the Sentinel system, the Sentinel Innovation Center focuses on developing innovative methods to further advance Sentinel by leveraging new data sources, building new tools, and enhancing real-world evidence capabilities through advanced analytics. At the Sentinel Operations Center, where I am, we focus on supporting FDA in the use of the Sentinel Common Data Model and its associated modular programs or tools to gather evidence to support regulatory decision-making. The Sentinel Operations Center maintains FDA's Active Risk Identification and Analysis System, or ARIA, as it's called. And together, we launched the webinar series to engage experts and innovators in key areas related to the Innovation Center's master plan and the Operations Center's methods work. And with that, I'm pleased to introduce today's speaker, Dr. Rishi Dessai, somebody who I work with very frequently. Dr. Dessai is an associate professor of medicine at Harvard Medical School and an epidemiologist in the Division of Pharmacoepidemiology and Pharmacoeconomics at Brigham and Women's Hospital. His research focuses on understanding the use of medications and resulting outcomes in routine care patients with chronic diseases. He has a special interest in methodological investigations to improve inference from non-randomized studies of medication effects, and he is the operations chief for the FDA Sentinel Innovation Center. The title of his presentation is A Process Guide for Inferential Studies Using Healthcare Data from Routine Clinical Practice to Evaluate Causal Effects of Drugs, what we call the Principled Project. Before we get started, I'd like to do a little housekeeping and remind everyone that their lines are muted. If you'd like to pose a question, please type it into the Zoom Q&A feature, and we'll be answering those questions at the end of the session. Finally, this session is being recorded, and it will be made available along with the slides through the Sentinel webpage, which can be found at www.sentinelinitiative.org. And so, without any further delay, let me ask Dr. Dessai to thank you for joining us and get started.
Great. Thank you, Dr. Morrow, for that kind introduction, and really excited to be here to present on this work that really has been a central part of our work at the Innovation Center over the last couple of years. And so happy to also share that before this presentation, the paper came out in the BMJ a couple of weeks ago. So in case you have interest in reading more about what I'm going to talk about today, it's all available freely online for you to browse. I also want to take an opportunity to thank everybody that is involved in shaping this paper. So this was, as you will see on the slide, this was a large undertaking of more than 20 co-authors and really gave us a lot of diverse viewpoints that we were able to kind of put together in this document, which really outlines a process for inferential studies that we are hoping to make the most use of in Sentinel moving forward. So just a note that this paper is available freely online on BMJ website. Okay, so the first few slides, I really wanted to build the case for why this is needed. What's the motivation behind this? And if you have been kind of someone following the literature in this space, you are likely aware of maybe all, but definitely some of these initiatives. So there are various frameworks out there, with some of them with in the recent years. And there are papers that provide you with tools to do quality assessment. So Robbins is an example where you have a checklist of sorts to assess how you can trust, or if you can trust a paper that is published using non-randomized data sources and methods. So this is a quality assessment tool. Then there are reporting tools out there. So Record PE is an example. There is the Stat RWE, which is a more recent example. So these are frameworks that allow you to prepare and transparently report all the operational details of your study. Then there are best practices guidance documents from the regulator. So EMA has the NSEP guidance for method standard in pharmacoepi. The FDA has some guidance on real-world data using the EHRs and claims. And these best practice guidance documents are very useful because they're very comprehensive. They're almost mini textbook for pharmacoepi and what researchers should strive to do. But they're highly descriptive and in a format that is very useful for people to get a comprehensive view of these concepts. And then there are some highly specific miscellaneous documents like Legend, for example, from the Odyssey community, which is highly tailored to the type of studies that they conduct. There's also other guidance documents out there like the SPIFT, for example, which is focused on part of the process, which is identification of data for conducting these types of studies. So when we were kind of at the beginning of this project in 2020 or so, what we realized is that there are various tools available for specific purposes. So there are tools for quality assessment, like Robin's Eye, for example. There are things for reporting, like START-RWE, for instance. There are best practices guidance documents, like the stuff from NSEP and the FDA. But what we don't have really is a general framework to guide decisions at various steps. And as we have kind of evolved in the field of pharmacoepidemiology, studies, we realize that there is a lot of thought that goes into designing a study, a high quality study. And if we can kind of provide people with some stepwise guidance on what are the considerations that should always be thought of when you are planning a study and implementing a study, we felt like that could be of great value to Sentinel. And for the scientific community in general. So the Innovation Center developed this causal inference framework, which proposed a stepwise process. It's really a process to consider all the key choices that you need to make when you're designing and analyzing the study. And all these choices are obviously directly related to the validity of the study that you conduct. And we are strictly focused on studies that are conducted using healthcare data, so that's EHRs and claims data, because that's what is of greatest relevance to Sentinel. And so the standardized process that we outline in this framework, we hope that it will serve as a guide to inform the conduct of these types of studies. And the key considerations as we were thinking of these steps and thinking of how to apply this for future Sentinel investigations, what we really wanted to do was to achieve a few things, which is listed in this slide on the last few bullet points. So we wanted to limit variations in practice. By no means we want to guide or make a process that is dictating choices for investigators. There has to be some kind of flexibility for case-by-case basis. But we wanted to set a baseline where everybody is considering similar things for their study, similar considerations. And that could lead to proper documentation of what a researcher must be thinking when they designed these types of studies. And so we wanted to limit variations in practice. Really, that's what this is. We also wanted to develop a process that was repeatable because, as you might imagine, Sentinel does many dozens of studies every year for looking at an inferential goal. Associating a medication with an outcome of interest. So we wanted a process that could be repeatable across these investigations. And we also wanted to invite independent experts, not just within the circle of people that we have always collaborated with through Sentinel, but also people who have written textbooks on the subject and who have put in a lot of thought throughout their career in this to make sure that what we produce is a cutting edge document that is reviewed and authored by independent experts. And we feel like with Principle, we were able to achieve all these things. And so I'm going to walk you, hopefully this provides you with a good set of considerations that we had when we were planning the principal process. So what I'm going to do in this presentation is going to walk you through all the overview of the process that we have described in this paper, and also walk you through each step in terms of how to think about these steps when you have an actual research question in mind or at hand. And so we have an example that we have in the paper as well, which is an example of a known drug event association with SGLT2 inhibitors and a risk of genital infections, which is a known adverse event. We wanted to keep something that is not very controversial, just because we were using this as an example in describing this larger process. But I think having an example make some of these concepts very, very tangible and more accessible to readers. So that's the hope today. So first, I'll introduce the process. So as I mentioned, this is a stepwise process guide. So this is the overview of the entire process. So there's a lot going on here. So I'll try to break it down in highlighting a few key things. So what we have here is the 5 steps on the left. These are the key steps that are always considered whenever you are doing an inferential study. Now, what you also will notice is that these are divided into the study planning phase and then an inference phase. So what we mean by that is that the steps 1 through 4 are all taken when you are planning the study. And at that stage, so you are not conducting the analysis in steps 1 through 4, because the reason is that you don't want to make changes in how you're doing the study based on the knowledge of the result. And so that's why we made a clear separation between the planning and the inference phase. Step 5 is dedicated just to conduct the analysis. And this will be clearer as I go through each of these steps, but just wanted to call this out right up front. You will also realize that there are specific paths that you can end up depending on the decisions or findings at each of these steps. And some of these findings will actually lead you to kind of enter the process again at a different stage to reconsider the question that you are asking, for example. So just wanted to kind of make these broad points that there are planning steps and there are inference steps, and then this process also has loops that allow you to go back and make principal changes. Okay, so having said that, now let's kind of introduce all these steps. What are these steps? So the step first is really asking the right question in the right manner. And what we have found useful is this target trial paradigm that has really gained a lot of steam over the last 5 to 7 years. So what this is really is a mechanism to ask a well-defined causal question. So the first step is specification of a target trial protocol. What this basically means is that you are envisioning your observational study or pharmacoepi study as if you were designing a prospective trial. And so the first step is specifying exactly what the question is that you are asking. So this is a fairly kind of intuitive starting point. Every time you want to understand something, you have to ask a question in a right manner. Now, from step 1 to step 2 is where you start kind of making the question more explicit by operationalizing all the details around it. So step 2A is where you kind of outline emulation of each of these components. So what this means is that you're basically explicitly saying how you want to operationalize all the specification steps that you've specified in step 1. Now you will realize that this process is kind of intertwined with the data that you have at hand. So that's our step 2B, where what you have is you have to decide if the data that you have at hand are able to support the emulation of the target trial that you want to emulate. And so because you are limited in some cases by the data that you have at hand to emulate certain components of a trial, this will dictate how you want to operationalize. And so that's why the arrows are going back and forth. So once you start emulating all the components of your target trial, you have to also go back to a data source that you have at hand and consider whether these types of measurements are available in the data set or not. And depending on what you find there, you may want to kind of make certain choices in terms of how you want to emulate the concept. And so that's why there is a back and forth between steps 2A and 2B, because we feel like these steps are intertwined. Okay, so hopefully that makes sense. So once you are through step 2, what we have next is that at the end of step 2, you will either find that you will have a fit for purpose data source that is allowing you to emulate the target trial that you want to emulate, which is the best outcome. And in that case, you can go on to register all your choices. So this is registration of the protocol, and it doesn't have to be formally registered in a repository like clinicaltrials.gov, but you have to have public dissemination component of it. It could be deposited on a website that you might have for a specific study or a specific program, for example. So the goal here is to really pre-specify all the choices that you are making. And by doing that, similar to trials where this is required before you start enrolling patients, the goal here is the same, is that you don't want to be in a situation where investigators are making changes based on what they find in the data. Although it's really easy to fall into that trap when you have the observational setting where you have all the data at your hand, you feel like a principled approach of doing this upfront would prevent a lot of or increase confidence in these types of studies. So that's the best outcome. However, in some cases, you will also find that the trial that you want to emulate actually is not supported by the data that you have at hand. And that is a situation where the fit for purpose data are just not available. So in that case, what we say is that you should stop right there and then reassess the question that you have, that you are asking. In some cases, what this means is that you may want to ask a slightly different question that is also of interest, but now that could be addressable by a data source that you have. Or the other approach is that you may want to go out and seek a different data source that could support the original question. So this is a step where we say stop and reassess. So that's the step 2. So assuming that things go well and you register your protocol at the end of step 2, then you move on to step 3. Now, step 3 is an important step in the process because this is where you are really making some inroads with respect to understanding the data that you have in relation to the study that you are designing. What we mean here is that you can make some basic diagnostic evaluations, as well as you can assess the expected precision of the analysis that you want to conduct. And this is really important because both of these things are quite useful to make principal study adaptations. And what we mean by that, and this will become clearer as I describe the step 3 in more detail using the example, what we mean here really is that there are situations where you can learn from the data that you have by these diagnostic evaluations, as well as by the precision evaluations, important and valuable information. And so if you feel like that this is a scenario where the desired precision is achievable and diagnostic criteria are met, then you can move on to step 4. But if not, then again, you are in a situation where you should stop and reassess. You can consider alternative design choices in step 2 that could increase the precision, for example, or that you could reassess the question that you are asking, whether the question is answerable by the design and the data source that you have at hand. So again, there are a couple of stopping points that we clearly outline in the process at step 2 and step 3. So assuming things go well at the end of step 3, you move on to step 4, which is really a way for you to kind of prepare for more robust inference, because here what we are saying is that you should also develop plans for robustness evaluation. So this is typically considered for most pharmacotherapy studies where you have a bunch of sensitivity analysis planned, and we feel like there is a lot of value in upfront pre-specifying the sensitivity analysis, not only the deterministic ones where you change one parameter at a time, but also the probabilistic ones. So for example, there are frequently the sensitivity analysis which are focused on unmeasured confounding. For instance, there are bias modeling approaches. If you pre-specify all your parameters for such a bias modeling exercise, it becomes really difficult for you to kind of game the system because all your choices are pre-specified at the outset. So what we are saying here is that if you decide to follow this framework and go through the steps 3 and 4, it is likely that your protocol that you registered at this point may have changed because of, or may have, because of potential study adaptations that you might make. So at this stage, it would be appropriate to log all these changes in an amended protocol. And I will go through an example where this will become clearer as well, but we feel like this is a really important step in designing observational studies where you can learn from the data that you have at hand and still stay true to the question that you're asking without being too subjective by the knowledge that you gain. But the protocol registration and protocol amendment steps make sure that this happens, because this is disseminated at various steps in the process, and because this is logged, there's no way for you to go back and make choices that are favorable in a way to the question or the answer that you want to see. So I think that this is hopefully making sense up to this point. But again, as I said, we will go through all the steps and hopefully that will clarify some of these concepts. So what we also say is that once you are through step 4, really a lot of hard yards have been covered. And step 5 becomes really easy then, because this is really when you are implementing the inferential analysis. So all the choices, the design choices, the analytic choices have been pre-specified up to this point through step 4. You also know the model diagnostics through step 3. You also know the expected precision through step 3. Step 5 only requires you to implement the final analysis to make the inference. So overall, I think this is a stepwise process that is applicable to really any inferential questions that you want to ask of healthcare databases. Now, in the next section of this talk, I will go through each of these steps one by one, and then cover the example that we used in the paper to make these concepts more tangible. Okay, so the step 1, as I said, this is the most intuitive step of all. Here is really an attempt to ask a well-defined causal question in this target trial framework. The example that we used here is the example of SGLT2 inhibitors versus DPP4 inhibitors, which are a separate class of medications for diabetes as well, second-line diabetes treatments. And the risk or the outcome of interest is genital infection. And as I said, this is a known association, so we did not want to study something that was controversial or unknown. So we picked this example on purpose. So what the specification of target trial protocol requires is that you specify the eligibility criteria. So these are your inclusion criteria, the treatment strategies that you are interested in comparing, the treatment assignment, the follow-up start and end, the outcome, and the causal contrast that you're interested in. So the specification column includes the choices that you want to make, and the emulation column is in some cases, you may not be able to kind of to achieve everything that you want to achieve in the specification step. You may need to modify this somewhat in terms of how you can operationalize this using real-world data sources. So that's a difference between the specification, which are really all the choices that you would ideally make, and the emulation is in many cases it would be same as the target trial, but in some cases you may want to make certain changes. So for example, the treatment assignment, obviously in a target trial specification, you would want to randomize them, but in the observational setting, there's no way for you to randomize. So you have to make this clear that this is non-randomized, non-blinded, and the assumption is that randomization is within the levels of measured confounder because you're adjusting for these confounders. So basically, the specification step allows you to explicitly state your causal question of interest, which in this case was SGLT2 inhibitors versus DPP4 inhibitors on the risk of genital infections. Okay. Now that allows you to move on to step 2A, which is the operational choices that you make to achieve the specified target trial protocol. So here what we are saying is that we can leverage actually a lot of work that has gone into this type of activities. So a structured protocol detailing operationalization of all the variable definitions, including all the codes that you're using to identify medical conditions for eligibility criteria, for treatment strategies, how you're defining them, outcomes confounders, everything can be highly detailed and operationalized in one of these structured templates that are available. So Start RWE is just used here as an example. There are others out there as well, including one called Harper, for instance. And the good thing about these protocol templates is that, again, they are there's a lot of thought that has gone into this already. And so you basically can leverage these existing templates to log all your choices in a way that makes them replicable, really. If anybody wanted to do the study again, they can easily find exact choices that you made in the databases that you had at your hand. So this is a really important step of the process, is transparently outlining all the operational details of your study. So then comes the step of identification of fit for purpose data. And so this is what I was saying, is that we did not make a separate step for this from 2 to 3. We made them 2A and 2B, the emulation and the fitness for purpose assessment of the data. The reason is that I think that these are intertwined processes, because sometimes you will need to go back and forth between the two. So this figure looks somewhat complex, so I'll try to go through this, but it really is an attempt to kind of make this consistent with the FDA guidance on the real world data that they put together for the industry, which was published a few years ago. So this guidance for real world data really emphasized two key things. So one was data relevance, and the other was data reliability. So data reliability is a concept that is kind of easier to conceptualize, which involves accuracy, completeness, provenance, and traceability of the data. And this is all upstream, where you, for Sentinel, this is all upstream, where you are converting your raw data into the Sentinel common data model. So we require a lot of quality assurance checks to ensure that all these reliability assessments are met for all the data that goes into Sentinel when we convert the raw data to Sentinel common. So that this one is an important one, but for Sentinel, this is something that we have always been doing. So we are very proud of that. For relevance, this is a more nuanced concept, because this requires the investigators to ask some basic questions, and based on the answers, you can go one way or the other. But what really I want to highlight here is that at the heart of the whole thing, there are 4 questions, basically Q1 through Q4, that relate to how well the concepts that you're interested in are measured in the data set that you have. So we start with insurance claims data in Sentinel, because that's the largest asset that we have. So we ask these questions of insurance claims data about the exposure of interest, eligibility criteria of interest, the outcome of interest, and the confounders of interest. So depending on the answers, you can stay within this box, or you may want to expand it out to incorporate additional sources. So I'll give you a few examples here. So for instance, if you are interested in a comparison of treatments for an indication like diabetes, you feel like there are good algorithms out there that can use ICD codes or combination of codes and drugs to identify diabetes with a high validity. And so the questions like this can be addressed where the eligibility criteria can be assessed appropriately just using the claims data alone, then you can stay within this box. However, if you have more nuanced conditions, so for instance, heart failure with ejection fraction subtypes. So heart failure is well recorded in claims data, but the ejection fraction subtypes is not. And as some of you may know, the treatment responses vary based on what subtype you have, whether reduced or preserved ejection fraction. So if you are in a situation like that, you may want to incorporate this additional sources. And the most obvious one that comes to mind to enhance the claims data framework are EHRs. So linkage to EHR in this situation could be handy for a few things. So you can use that linked claims EHR data, which will be for a subset in most cases. You can develop and validate claims-based algorithms to see if the eligibility criteria that you're interested in can be identified, not using just the ICD codes alone, but using some kind of probabilistic phenotyping algorithm, for example. And if you see that that is something that is achievable, you can actually take this algorithm and go back to the claims data source where you have the advantage of size. And you can apply this algorithm that you developed and tested, and in some cases this may be pre-existing, and you can apply it to the larger population to identify the condition that you are interested in. However, in some cases, you will find that the algorithm is not demonstrating adequate performance, is not able to identify the condition of your interest. In that situation, what we say is that you may want to restrict the entire study to a linked population alone. This comes at a cost of compromising the size of the study. However, if there is no way for you to reliably identify the central components of your study, we feel like you should probably focus on internal validity at that stage and conduct a study in a more restricted population. So this is the basic way in which you can read this chart. There are also similar considerations for outcomes, for instance, treatment, as well as key confounders. And depending on how you answer these questions, you can stay in the box or you can incorporate additional sources. And depending on each case-by-case basis, you may find that the insurance claims data alone or claims data augmented with some other sources are in combination relevant for the question that you're asking. So this is all under data relevance assessment. We also have this box for initial feasibility assessment, which is allowed for data relevance checking. So this includes things, very basic checks like how many new users of a medication that you're interested in are in the data set. So you're really not asking any specific questions, but just basic feasibility questions, whether this data set is relevant to identify the exposure outcome interest that you are interested in. So based on this exercise, you will hopefully find a data source that or combination of sources that is relevant as well as reliable. And when you meet these two criteria, we call these data sources fit for purpose, which is entirely consistent with how the FDA describes this in the real world data guidance that they put out for the industry, which was one of the goals to keep it consistent. So that's our step 2B. So in the example that we had of this SGLT2 inhibitors, we asked these questions. So can the eligibility criteria be emulated? We said yes, type 2 diabetes has a high PPV. The outcome of interest, genital infection, was also identifiable with a high PPV based on published studies. And then treatment, of course, we are only interested in the outpatient prescriptions, which are well captured in claims data. The question of confounders was an interesting one in this one because we felt like genital infection had many risk factors, including the control of diabetes, which is not as well measured in claims data. So for that, you may want to consider linkage to EHRs, where you have information like HbA1c, which allows you to control for that, how well the diabetes is managed in the patients. And you derive this additional information on unmeasured confounder that are unmeasured in claims data. And then you might use this information to either conduct supplementary analysis, keeping the main analysis claims data, to preserve the size, or you may want to restrict this in the linkage box if you feel like that this confounder is really something that you have to have. in order to gain confidence or trust in the study. We also conducted some initial feasibility checks, which allows us to look at how many patients there are new users of these medications in our study population were. And as I said, the data reliability assessment is upstream for all the data sources that are contributing to Sentinel, because this is taken care of when we are going from raw data to SCDM, the common data model. So in this specific case, what we said was that we answered yes to the first three questions, just claims data alone. However, for the key confounders, we felt like information such as HbA1c could be quite useful for controlling confounding, and that's the reason why we decided to use an asset that was linking claims data to EHR, which was the EHR from our hospital system, the Master Brigham hospital system, linked to Medicare claims. So this is the linked asset that we said is fit for purpose for studying this question. So this is just an example of how you would walk through this process when you have an actual study question in hand. So that allows us to move to step 3, which is some guidance on conducting precision evaluations as well as diagnostics. So this step 3 also has steps 3A and 3B. So 3A is entirely based on precision. So what we mean here is that based on the outcome counts in the overall study population, not stratified by treatment, you can conduct preliminary investigations under some assumptions of the expected precision of your eventual results. You can also conduct basic diagnostic evaluations. So for instance, some general diagnostic evaluations that we always conduct, which allow you to look at the baseline characteristics in the treatment in the reference group. There are also some analysis-specific ones. So for example, if you're using propensity scores for confounding adjustment, you might want to look for the adequacy of the PS model that you build through evaluation of covariate balance, for instance. If you're using something more complex, like the censoring weights, for example, you may also want to look at the distribution of weights. So there are analysis-specific diagnostic evaluations that you may want to do, but there are some general ones that you may always want to do. So based on the expected precision calculations and diagnostic evaluations, assuming that everything checks out, you may want to proceed to the next step. However, there may be situations in which the desired precision is not achievable, or there are potential issues that are not addressable by refining models. You may want to go back to step 2 and consider design modifications, which is what ended up happening in the example that I was describing, which I'll summarize in a second. Okay, so this is for the case example now. So as I said, the data source that we felt like was fit for purpose was a data source that was combining claims data and EHR from the MGB system for this research question of interest. So we went ahead and conducted the precision evaluation in step 3A, so which requires us to implement all the eligibility criteria and assign eligible individuals to treatment groups. So we implemented all the inclusion criteria, which are here in this flowchart, and we saw that at the conclusion of implementation of all the inclusion criteria, we had about 1,500 patients in our study cohort, which were divided into 1,000 patients in the DB4 group, which is the reference group, and SGLT2, about 500 patients. So just based on these counts, we looked for the overall number of outcomes in the study population, not stratified by treatment groups. So that's the number that you see here, 40. So that's out of 1,500 patients, 40 of them had the outcome event. And based on some assumptions of null treatment effect, for example, you could have any treatment effect, you could calculate using standard formulas that are available. And we also provide an R code to do that along with the paper to get at the expected confidence interval. So this confidence interval, based on the number of outcomes that we had here, we calculated it to be 0.35 to 1.65. So this basically was an indication for us that this likely is not sufficient sample to arrive at a treatment effect that is precise enough for us to make a conclusion about the potential association between the drug and the outcome. And so we said this desired precision is not available, so we would go back to step 2 and consider some design modifications in this study to achieve better precision. And that's what we end up doing. So in the revised version of the targeted protocol, what we ended up doing is we basically saw that if you required people to have HbA1c test results available, you dropped a lot of patients in there. So from 9,000, you go to 1,400. So we decided that that's a trade-off that we were not willing to make, which is that dropping so many patients just because we could have access to their HbA1c test results. So we decided to stick to or drop that inclusion criteria, and doing so made 293 events available to us. And this translated into a confidence interval of 0.73 to 1.27, which compared quite favorably with what we had before, which was 0.35 to 1.65. So we felt like this was a decision that we had to make, a trade-off that we had to make in terms of gaining the precision at the expense of the more granular information on HbA1c, which still remains a useful confounder and which we have to account for in some way, just not directly by restricting on the availability of results for those patients. So after this modification, for this case example, we felt like desired precision was achievable, and so we proceeded to step 4. And I'll describe how we addressed the HbA1c issue in a second as well. So step 4 has to do with robustness evaluations, and we have some guidance on the type of robustness evaluations that you should consider. So this is divided into deterministic, probabilistic, and net bias assessments. So deterministic is something that we always consider weighting assumptions one at a time, maybe changing the outcome definition, for example, something like that. The probabilistic ones are also frequently considered for exposure outcome misclassification, for unmeasured confounding. And then there are these net bias assessment approaches that are, I think, up and coming. I don't think this is routine practice yet, but I think it will be at some point in time. There are various kind of blunt tools available for this. So you have the control analysis, negative positive control outcomes. If you can replicate a known association there, or non-association, you may have slightly more increased confidence in your primary results where the association is not known. The trial calibration and benchmarking is relatively new, which is an idea where you may want to look at a well-conducted trial and replicate the entire trial using exact inclusion-exclusion criteria and replicate the primary study results for the trial. So if you are able to align your results for a different outcome, perhaps, or for a different population, using the data that you have at hand, using identical inclusion-exclusion criteria and design choices, you may have slightly more increased confidence in the unknown results that you are pursuing, maybe a different outcome or maybe a larger population. This approach, I have to say, is quite limited because there are very few instances where such a trial is available, comparing the exact two groups that you're interested in, that's what makes it really tough to pull off, but not impossible. We have done this in the past, but we recognize that this is a really a question of availability of such a trial. Okay, so what we ended up doing for this specific case example is that we outlined a bunch of deterministic sensitivity analysis, which are outlined here. So we had a more specific outcome definition. We varied the follow-up scheme, and there were probabilistic sensitivity analysis, which is an important point here. So if you recall, a few slides ago, I was talking about HbA1c and how we ended up dropping that inclusion criteria that required people to have HbA1c test results. However, we still had availability of HbA1c test results for the 1,500 patients that we had who had results available, which was a subset of the overall 9,300 patients that we ended up including in the study. So what we did here is that we used the EHR-linked cohort to inform our assumptions for the probabilistic sensitivity analysis for unmeasured confounding. And I think this is a really important activity that you can undertake, which is based on real information that you have, as opposed to assumptions that you make routinely for conducting these types of analysis. So having availability of the added information from EHRs allows you to make these types of informed choices when you are conducting your robustness evaluations. So we ended up doing that. And for the net bias analysis, we did not have an exact trial that we could replicate, but we did know that hospitalization for heart failure is a known positive outcome that SGLT2 inhibitors are associated with, but not the DPP4 inhibitors. So we use that as a positive control. So just to replicate what's known in order to increase our confidence in the unknown. Okay, so that's the steps 1 through 4 and how we implemented them in our study, which left us with just the last step, because everything is pre-specified now. We have all the machinery ready to go. We have all the robustness evaluations that are specified. We have all the models that are evaluated through the model diagnostics in step 3. So all we have to do at step 5 is implement the final analysis. And this is how we ended up doing that. So this is the primary analysis for the full cohort for all the pre-specified subgroups, as well as all the robustness evaluations. And as you will see here, you can find a consistent association that is, as I said, known between the genital infections and the SGLT2 inhibitors of a similar magnitude across most of these analyses. And for the positive control, we were also able to replicate the known signal for the heart failure outcome, which is a good kind of reassurance that we are able to address or retrieve the true result in that case. You will also see that we also present the robustness evaluations results, so sensitivity analysis and the quantitative bias analysis in the same table, which are all pre-specified. And then the QBA, I wanted to highlight a bit more because this is correcting for unmeasured HbA1c. And the uniqueness here is that the HbA1c information we have available for a subset, which allowed us to make these assumptions more real life. So what this is a 3-way graph from this. This is using methods by Flanders Corey, but there are many such methods available out there that you can use off the shelf. But what this is essentially saying is that depending on the combination of the prevalence of your unmeasured confounder as well as the exposure unmeasured confounder association, you can recalculate the adjusted relative risk. So what this is useful for is that if the adjusted relative risk shifts quite a bit, then it tells you that the unmeasured confounder actually potentially has a large role in explaining the results that you are seeing. But in this case, what we found really was that we started off with the bias parameters that are observed in the ancillary data. So that was 14% prevalence of the uncontrolled hyperglycemia, and then the odds ratio of 1.3 for SGLT2. So these are the two parameters on these two axes. And then we calculated the corresponding relative risk, which came out to be 2.63, not very different from the 2.68 that we have as the primary result. And then what we said that this is the base case. This is what we see in the ancillary data. Now we are also going to assume that what we see in ancillary data is potentially the best case scenario. There are associations between the unmeasured confounder exposure that are larger, or unmeasured confounder and the disease or the outcome features that are larger out there. You just happen to see this really muted association in the ancillary data. So everything else that you see here is making these assumptions stronger regarding the confounding triangle. And what we see is that even if you make them extreme, so that's the 24% uncontrolled hyperglycemia, which was only 14% in the ancillary data, and the odds ratio, which was 1.3, if you make it 2.0, even then the adjusted relative risk only goes down to 2.48 compared to 2.68 that you have in the main results. So this is, I think, a useful way to construct or think about the bias evaluations, the bias modeling approaches for unmeasured confounding by basing your assumptions on observed data in a subset. So which is what we ended up doing for this study in lieu of adjusting for it for everybody, because we were not able to do that. We had to make that trade-off for achieving precise results, as I was describing in step 3. So that's really the last step. This was the result from our inferential analysis, and I wanted to kind of summarize the whole activity by saying that the stepwise process systematically considers key choices and decision nodes whenever you're considering an inferential study. And we hope that the process that we outline here will obviously be useful for Sentinel moving forward. And we are actually testing it out for a couple of demonstration projects as we speak. But we also think that this could be a useful framework for people outside Sentinel to facilitate communications between various stakeholders and motivate critical considerations for clinical research. And ultimately, the goal here is to increase confidence in and allow people to conduct studies that are hopefully reproducible and transparent in all the assumptions that they make, which eventually would allow for increased transparency and confidence in these results. So that was my last slide. I think I'm happy to take any questions, but really wanted to thank you all for attending this today, and hopefully you will take a look at the paper as well.
Thank you, Rishi. So grateful to have you come and speak with all of us today. There's some questions in the Q&A, so I think we'll go ahead and transition to the Q&A portion of the webinar. Rishi, I think it might be useful if you actually went back to the visual display of the 5 steps for some of these questions, because I think it will be easier to answer them in that context. So a couple of these questions start out asking about the order of things and whether there is an importance in, for example, putting the inference after the sensitivity analysis, probabilistic sensitivity analysis and bias planning, or whether there should be maybe 2 inferential steps, one after the primary analyses are complete, and then sort of thinking of those as auxiliary or sensitivities. Can you speak to what you feel about those things?
Yeah, so that's a really important question. And I would like to answer that by starting out with a statement, which is that we don't think that this is the only process that is acceptable. We feel like this is a useful set of considerations that are always kind of at the back of a researcher's mind when they are investigating these types of questions. There may be certain choices that you may have to make that differ from this, which is totally fine. And we say this explicitly in the paper as well. Coming back to the specific question of where does the sensitivity analysis package kind of fits into this process. And we actually went back and forth on this quite a bit. And we had strong opinions on both sides. We ended up picking this before the inference step because we felt like what we see routinely as kind of stepping aside for a minute in my academic hat as a peer reviewer, what we see routinely is that people see a result in the primary analysis, and then they, based on the results, it's easy to kind of come up with assumptions that eventually would lead to a conclusion that these results are robust to any of these problems, right? So it's a really simple arithmetic at the end of the day, the bias analysis pieces, and you can basically make up these assumptions to make your results look like they're robust. So we felt, or at least more of us felt that pre-specifying some of these assumptions is the way to go here to minimize subjectivity in reporting and conduct of these sensitivity analysis. But we by no means are saying that this is the only way to do it. I mean, there are potential things that you discover post hoc that would lead to more sensitivity analysis, and I think those are all useful. It's just that you should put limited stock in interpretation of those if they are not pre-specific.
Yeah, thanks, Rishi. There's another set of questions that is trying to, I think, put all of these frameworks into context, and particularly the relationship between existing frameworks. And so there's a reference here to looking at steps 1 and 2 here. So these are the components, where you're ultimately getting to fit for purposeness, and asking if there's anything that's really different about these from the existing SPACE or SPI-FD frameworks, or if they're largely covered in these first two steps in those frameworks as well.
Yeah, so I think the central considerations, I'm not going to claim that we invented something new here. I think what's new here really is taking a step back and think of the whole process. So as much as we like to think about fitness for purpose, it's just a piece of the puzzle, right? So I think we have to think about these steps as intertwined and as kind of dependent in achieving a larger goal of conducting the study and eventually really answering the question that is of interest to the stakeholders. So I don't think there's anything new here per se. The target trial framework, for example, has been described maybe 100 times now. But what we are doing here is we are really putting pieces together to come up with a process that hopefully is easy for people to see that how everything interacts with, how all the decisions interact with each other in pursuit of this larger goal. So I would say that you will find pieces of this in many frameworks outside of this framework. And some of them we actually explicitly rely on them, like the START RWE or Harper, for example. That's our step 2A. We did not spend any time thinking about that. That's likely because that work was already done. So we are kind of stepping on the shoulders of giants, I guess. But that was a really good question. Thank you for whoever asked that question.
Yeah. Folks, I don't see any more in the Q&A, so I encourage you, audience members, to continue to ask questions. We have just a few more minutes. I'll take the moderator's privilege of asking a question and just say, Rishi, your example here is one that we are now starting to see just repeat itself over and over again, which is this issue of how much of the subsample do you need to be able to check your assumptions on the study? Because we're seeing it in a wide variety of our feasibility assessments where we do have some laboratory results data, maybe some BMI and smoking data, but it's really in a subset. And I think, do you have any advice for how small is too small? Would it be, is 5% of the sample just not worth bothering with? Or, do you have any feelings of threshold for how much you need in order to provide that sort of extra reassurance, doing some of the methods that you talked about here to check some assumptions and things like that?
Yeah, so that's actually the million-dollar question, right? So we all want to achieve a state where we have everything for 100% of the sample, but that's never going to be the case. It's not just limitations of the data source, it's also a reflection of the healthcare system, right? So many times these tests are just not ordered or patients just don't bother getting them. And so it's really a complex issue of the data generating process itself. So that's not directly answering the question, but just kind of sympathizing with your troubles there. To address your question, I think any information is better than no information is my take on this. You have multiple ways to approach this in terms of how sophisticated or how many assumptions you want to make in terms of correcting for these types of biases. The approach that we took here is actually not making many assumptions because we are modeling out many scenarios. We fully recognizing that what we see in this subset is likely some version of truth, not the entire truth. And so we recognize that, and we model out a bunch of other scenarios as well. So this is on the spectrum of making fewer assumptions. On the other end, you have methods like subset calibration, for example, which and these are all active areas of investigations, I have to say, in the Sentinel Innovation Center, because we want to provide tools that can help really instead of just preaching that you should do this and that. So if you are intending to use methods like that, it should come with a recognition that there are many assumptions that are involved there, and the fewer the data points that you have, it's harder for you to make those assumptions or believe those assumptions. It will spit out a result. It's just how much confidence you have in it. So I would say that over the next 3 to 5 years, just from the Sentinel Innovation Center, I think there are going to be substantial amount of results coming out, providing guidance on some of these things.
All right. Well, I want to thank again Dr. Dessai for presenting today's webinar. As a reminder, This session has been recorded and will be made available through the Sentinel webpage, both the slides and the recording. You can also find details and registration links for future Innovation Center and methods webinars and seminars on the website. And this will conclude our session today. I hope that we see you again next time. Bye, everyone.
Thanks, Judy.